Um, so we'll get started then. So before I begin, I want to acknowledge that the Field Museum resides on the ancestral homeland of Native peoples, including the Ojibwe, Odawa, and Potawatomi. We extend our gratitude and respect to those living here today, as well as their ancestors. And so it is my pleasure to introduce today's speaker, Dr. Joe Milanovich, who is currently an associate professor in the Department of Biology at Loyola University, Chicago. Joe's work focuses on understanding the effect of land use and climate change on and the ecological role of communities, both in natural and in urban environments. In doing this, Joe frequently is using reptile and amphibian communities to address his questions, and he has a lot of research that pertains to this occurring right around Chicago. So this is actually a really great place for this kind of work. I had the pleasure of first meeting Joe when I interviewed for this job that I have right here, right now, because he was on the search committee. And so given that I'm hosting him now, I hope he voted for me. It's like, yeah, maybe. And since then, I've had the pleasure to not just interact with Joe as a research associate here in Chicago and giving a talk up at Loyola, but also with some of the students he's taught who have turned out to be some of the best interns that I have ever worked with. So not only is Joe publishing great research around Chicago, but he's also inspiring and mentoring the next generation of natural historians as well. And so without further ado, I'll let Joe tell us more about some of the local work he's doing on aquatic turtles around here. Take it away, Joe.
Thank you. Everyone, thank you. It's really an honor to be here. Thank you, Sarah. Put quotation marks on everything she said. That's all debatable. I wanted to first thank everyone that did the actual work. So there's a host of crew from Loyola. Most of those are undergraduate students and graduate students. Jess is here today. I'll highlight them a little bit more as we go through. And then there's a crew from Benedictine University, mainly Dr. Leanne Harden. She's a faculty member there, a colleague of mine, a former postdoc of mine. And I would say that we kind of jointly created and conducted and designed this work together. So she gets a big shout out with her students, and some folks at the Forest Preserve districts of DuPage and Lake County were really instrumental in a lot of this work as well. And then our funding agencies are kind of varied from the DNR. We got some private funding and some university funding as well. So it was really a collaborative effort that I get the pleasure to boast about as I sat in my ivory tower in my office and watch kids track turtles. So when we think about turtles, I view them as this overlooked, really important piece of the ecological puzzle. If you look at them, they're really common. You see them everywhere. They're in most habitats, and they're super abundant where they are. And you don't comprehend the abundance of them. They rival fish in a lot of cases in biomass. And with this kind of combination of being widespread and really abundant, living in a variety of habitats, we kind of miss, I think, the importance of turtles in ecological function and in the importance of managing them and trying to encapsulate, what they do for ecosystems. And this is really prominent because they're in decline. Like most other vertebrates, turtles are globally imperiled. More than half the species are threatened. And as we'll talk about in the Midwest, most of the species in the Midwest are threatened. And when we think about the threats, there's the typical threats of land use change and land use conversion. There's a whole separate section with turtles where they're harvested pretty significantly. Millions of turtles a year are harvested just from North America alone, which we kind of overlook that as well. And then when you think about other aspects of turtle ecology, road mortality is really large, combined with this kind of land use change. So there's a host of threats that they have that's making them decline. Whit Gibbons is a famous herpetologist from Georgia that has done some work on this, trying to encapsulate this issue of turtle decline that again is often overlooked. When we look at it more broadly, you can see that this map is showing you the turtle richness across the world. And what's unique is that the Southeast US, and it dips into the Midwest a little bit, has really high turtle diversity despite this lack of decline. And when we think about turtles at risk, we see that almost half of them globally and in the United States are at risk in some capacity, either in decline or threatened or endangered. And what we'll talk about today are these Midwestern turtles. All these turtles have these similar traits of life history that are not unique to turtles, but that really exemplify why they're in decline. A lot of them are long-lived species. They take forever to become fecund, and they grow super slow. So when you look at land use threats and threats of road mortality and all these other issues, if you take forever to become fecund and you're a super slow grower, and then finally you can start laying eggs, it puts all this pressure on these populations. And when you have habitat that isn't of great quality or these populations are disjunct, it further exemplifies this issue of why they're declined. And what we see in the Midwest in particular is these populations that are old. You have old turtles that are outside of their reproductive age, and there's not a whole lot of recruitment. So even though we have these endangered species that we'll talk about in Illinois, a lot of these disjunct populations where they exist, they're not really viable. So we have to do mechanisms and different management techniques to try to implement this recruitment, which makes things interesting, especially in the Midwest. So most of the Midwest species, as we'll talk about, are kind of prairie adapted, I would say. And we blew down all the prairies for the most part. So that further exemplifies this issue. And then when you have these turtles that live in these remnant or restored prairies, they are generally surrounded by urbanization and roads, which weaves in this other issue. They still get harvested. There's a crap ton of pollutants. And then there's a bunch of predators because this mesocarnivore, effectively raccoons, have this huge threat to the turtles that exist in these populations because they love urbanization and roads. So they have all these pressures against them, and still they're quite understudied. This is a Blandings turtle range, and what you see is the green is the historic range of the Blandings turtle. They're really a northern Midwestern turtle, and they're currently endangered. All those red dots are where they live, but most of those red dots, as you can see, some of them are pretty far from each other, so they're naturally disjunct. You say, yeah, I can see how those aren't meeting up with each other. Even the ones in the Chicago area, if you look, they're close together, but they're separated by pretty significant urbanization or agriculture. So they are disjunct still, even though geographically the turtles theoretically could get to each other to interbreed and to mix. They can't. They'll get walloped by a road, or the habitat's unsuitable for them to get through. So we see this same sequence for all the three turtle species we'll see, is that they exist, they're threatened and declined, and they're all living in these disjunct populations basically by themselves. So the three we'll talk about are in some capacity either at risk or threatened or endangered. The ornate box turtle is a terrestrial species that lives historically in prairies in the Midwest and in the western kind of half of the US, Southwest. And they're in decline pretty significantly. I don't think we have enough data on them in particular. They're state threatened, at least in Illinois, most likely endangered. Again, it's data deficiency. And they're really a prairie adapted species. The Blandings turtle is an endangered species of the IUCN Red List. They're also this prairie adapted species, but they're semi-aquatic, and they live mostly in wetlands. They'll breed in these, kind of riparian areas. And historically, when the habitat was available, they would truck it. They would truck it miles to new wetlands. But now they're stuck in these little disjunct populations, so they have some issues. Spotted turtles are another endangered species that live in Illinois and in Indiana. They're more of a mix of a prairie forested adapted species. Their range goes up to Massachusetts and down to South Carolina and Georgia, and they kind of have this mixed variety habitat. They're also semi-aquatic, and like the other two, they're also in pretty steep decline. So what our objectives were, we had several projects going on for each of these species, and the talk is broken up into two different sections. We use these turtles to try to provide management data. So the DNR actually manages these disjunct populations in the habitat of these turtles. So the first section is where do they go? What are they doing? What sort of habitats are they using? And we weaved in this idea of looking at their thermal ecology, because most of these habitats that they live, especially in Illinois, are managed pretty heavily with prescribed burns and mowing, and they were killing turtles. So if we can tell them thermally the temperature is this, they start going down for hibernation, it's safe to burn, that really does help the management of these turtle populations. So the first half is just this kind of what are they doing, when are they moving, what's their thermal ecology, and the second half was really Leanne's talking points, which were using conservation physiology to kind of quantify the health and the stress of these turtle populations, just to get a sense of if they're healthy. So most of this is kind of baseline work, especially with the box turtle. There's really no work done with home ranges of box turtles in Illinois. There's definitely no physiological work at all in the species. So we use this as kind of a baseline project to get a sense of how healthy they were and provide some just values of what their physiology looked like. When you got to the Blandings turtle, it was a little more different because they're so disjunct and in such decline that all of the populations in Illinois are actually, well, I'd say most of them are from head-started individuals. So folks will collect eggs, they'll raise them in captivity for 1 or 2 years, and then release these hatchlings, really kind of 2-year-olds, into the wetlands. So most of the populations of Blandings, especially in and around Chicago, are very heavily managed with respect to head starting data and thousands of turtles being released. But there was still this huge lack of data on if they were healthy turtles, how do they compare to the ones that were born in the wild, and none of this was really looked at. The third species, the spotted turtles, we again collected the baseline of these kind of home ranges and the physiology. And then my grad student Jess will talk about more, weaved in this aspect of heavy metals. They're populations from Gary, Indiana. You can imagine what sort of threats they have there. One of them in particular was this heavy metal loads that were deposited into these landscapes. So we kind of looked at this from the angle of if these heavy metals are influencing the physiology or stress of these turtles in these populations. I'll quickly go over the methods here. For the most part, for every turtle species, we conducted ground-based radio telemetry. That's why we have so many students, because this is really difficult work. You glue transmitters on and some eye buttons, and you got to track them 1, 2, 3 times a week continually, so you can get enough GPS locations, so you can calculate these home ranges. Satellite transmitters are great. Those are for sea turtles where the money is flush, not for ornate box turtles where you're working off like 50 grand or less. Then for each one, we conducted a series of home range analyses. The minimum convex polygon is just basically a polygon around where all the points are. And the kernel density home ranges are a little more statistically focused, where they look at where the most points are located, and they do some statistical analyses to calculate a home range based on that. And then for each one of these, we used a series of mixed effects ANOVAs that allowed us to look at whether or not site or season or different turtle measures of, let's say, heavy metals or head started or not, if those had any influence on the home range sizes. Physiologically, we use this conservation physiology approach, and this is a really kind of emerging field of using this combination of morphometrics, where you can calculate the body condition of these species, and then this host of blood chemistry variables along with hematology. And you can put it all together and you can get this picture, this kind of outlook on the health and the stress and the kind of physiological health of these turtle species. And it's kind of a new emerging approach that really started with the sea turtle folks that had a bunch of money, and we kind of stole it and applied it to these terrestrial and semi-aquatic freshwater turtles. The sequence is pretty typical. We collect blood periodically. So in our case, we were lucky because we had these turtles that had radio transmitters, so we can find them. The students would dig them up out of the mud or wherever they were, and we can then take blood. And we didn't need a lot of blood because we use this really unique tool, this iStat auto analyzer, that needs a drop or two of blood, and it can run a host of these physiological metrics on site. It's a little handheld device. If anyone wants to use it, it's in my lab. We paid a lot for it. Use it all you want. And then we can make blood slides, and we did this for Blandings and spotted turtles. We made blood slides so we could look at this ratio of red to white blood cells and different types of white blood cells. And it can give us this long-term indication of stress in vertebrates. This is a really neat, unique technique. And then Leanne built a bunch of fancy linear mixed models to examine the differences of these physiological metrics and the hematology, and ask questions on whether or not it's influenced by temperature, the site that they're on, and all these different cool things. So what you'll see as we move forward is each of the 3 turtle species on top. And if it's red, that's the one we're talking about, because the data is kind of contiguous from each, but there's some small differences here. So for the home range work, we worked in Will and Grundy County, just outside of the city. We did 2 sites for 2 years. Really, this is spearheaded by my graduate student, Brock, my former grad student, who did this outside of his thesis work, which is impressive, and he published all of it. So Brock, with the help of the turtle dogs there that you can see in this picture, rounded up a bunch of box turtles. And if you're not familiar with the turtle dogs, this is John Rucker to the left there. He's a Southern former school teacher that literally travels around the country in a 15-passenger van that's converted with dog crates. And for months on end, he starts in the south, works his way north. He travels across the country, and you pay him to bring his Boykin Spaniels out. And he doesn't say they're trained, but they will go find turtles and they bite them and carry them and drop them at John's feet. And they're really unique, and John's really unique. You can imagine they all have names like Jenny Wren and Scooter and all these Southern names. And they're really great at finding these ornate box turtles in particular. So he has made a living off this for decades now. And he lives in Montana and just takes this van around. So the turtle dogs helped us. We were able to get these transmitters on the turtles. And then after collecting the data, Brock was able to calculate the convex polygon home ranges and some other metrics, which is pretty cool. So what we saw was pretty interesting. This is just one site with one of the home range metrics. We don't need to see them all because they're pretty congruous across years and across sites. But with these ornate box turtles, they effectively started heffing it right when the spring happened. They would emerge from hibernation. Their home ranges would get really big. That follows this general sequence of when they're breeding and when they're trying to bulk up for the summer. And then what we saw in almost every home range case was that the sex of the turtle didn't matter. You'd figure the males would be heffing it longer or more robustly to try to find females, but sex didn't have a big influence on the home ranges. What really did was the seasons. They slowed down the summer, they slowed down extra in the fall, and they were just kind of congregating around these brumation sites, and they did more or less this kind of communal brumation. And we were able to kind of calculate where and what types of habitat and stuff that the DNR could use. But what was interesting is we think that these home ranges weren't different across sexes in these populations. They effectively had nowhere else to go. They're prairie adapted species. There's a contiguous prairie where we studied them, and then outside of that was agriculture, either forests, which they don't use, or urbanization. So they really had nowhere else to go. So we saw the home ranges were similar to other species of, or other populations of ornate box turtles, but if you go out west, they're bigger. They can be bigger in the spring because they have more room to kind of round out. But we saw that we saw the seasonal pattern that was pretty clear. They start heffing it in the spring, and then they just shrink as it gets too hot, and they shrink as they start going under brumation. When we started looking at the thermal data, this is when it got pretty cool. What you're seeing here is the turtles are in the dark color, and all we did in this case was ambient temperature is the yellow, and the line going across there, that's freezing. And you can see that these turtles are really good at hovering around freezing. The lowest temperature recorded is 0.05 Celsius. So this brought up a lot of really interesting things. We can see where their brumation sites were. We can locate the hole. We can still track them under the soil, but we didn't quite know how they were doing that, if they were moving up and down the soil column to try to match the temperature. But whatever they were doing, they were hovering around that 0 Celsius, which is cool. And to us, what this signified was really that they were doing what they're supposed to do effectively. So even though they're disjunct, they're an older population of turtles, they had their groove on, which is pretty cool. And what the DNR really liked was we were able to quantify this thermal data to give them a temperature range of when they start emerging and ascending into hibernation. So then they can conduct their burns, because normal folks would say, okay, they go down in November. Well, what the hell is the DNR going to do with that? But if we could say once it hits 20 Celsius or whatever it is, they start slowing down, then they can use that no matter what year it is, no matter what the climate is, how it changes. And it resulted in them redeveloping their management plans for these prairies so they kill less turtles, which I think was pretty cool. So when we look at the Blandings turtles, again, these are the turtles that were head started. And we had this unique opportunity to compare these turtles with some that were formerly released, that were there for a little while. We couldn't do it with the home range analysis, but this was all really led by my student Armand. He was a grad student in my lab. He's at US Fish and Wildlife in Chicago now. And he spent several years looking at this site of these Blandings turtles. And each year he would release a new batch of these turtles, 10 or 12 that were raised in captivity. And then Peggy Noterbot took them, put them on display for a year. And then we grabbed them out of the little tank and just dropped them off in these wetlands. And you can see the transmitter and the I button there. And Armand was able to calculate a host of kind of kernel densities and convex polygons just to see if these brand new head started turtles were functioning similarly to what we'd expect, and if they're surviving. So he's able to get some really cool survival estimates. What Armand saw was pretty different. This is a semi-aquatic turtle, not a terrestrial one like the ornate box turtles. And Armand saw seasonal differences that was not unlike these box turtles that we saw, but it was kind of flipped. We released them in the spring. You can see the home range estimate down at the bottom there. We released them in March or April, and they started branching out a little bit. But once the summer hit, in each year across these different cohorts, we saw their home range really expanded pretty significantly. Each year they did this. And then once you get to the fall, like any other turtle, even though they're raised in captivity, they started congregating into these brumation sites. And what was unique is that we found these kind of communal brumation sites. That was pretty cool. Far from where they were released, they found the habitat they needed. It was just this innateness to them that was really encouraging to see. So we're happy to see these head started turtles do well with respect to their kind of behavioral ecology. When we looked at this a little closer, what we realized with the box turtles is that ambient temperature is like 1/10 of what you really need to do. So we buried eye buttons, these temperature loggers, into the ground at different depths, so we can try to see if the turtles are following these different depths, if they're having this kind of active brumation period where they're moving up and down the soil column or the water or air or whatever it is. And when you look at this, what Armand ended up doing is he kept the turtles in the dark color there. And then all those different depths are associated with these different temperatures across the months. And if you look at the bottom, you can see those are really those temperatures that are at the surface. So at 0 centimeters, that's basically the water. And then 15 centimeters is ambient. So the turtles were burrowing down. And it appeared that they would have this kind of active brumation. They're under the ice in the wetland, but they're in the soil of that wetland. And they would move down or up depending on how warm that soil was. Again, head started turtles there's no hibernation phase when they're head started. You put them out in the wild and they don't freeze. They hover right around that 0.05 Celsius somehow. And most of them made it through this brumation period, which is pretty cool. And again, what also helped us was that we were able to give some data on when they're brumating and when they're ascending from this brumation. So the DNR and these other agencies that are managing these populations were able to work their management plans around this. So it was pretty cool to see. So what Jess's work focused on then were these spotted turtle populations in Gary, Indiana. Again, there's two populations. She did it for a year, and they were incredibly disjunct. I-90, every type of industrial sites you can imagine were surrounding this. So they didn't have a ton of places to go for the most part. But what Jess was able to do is weave this analysis of heavy metals. These were native turtles. They were born there, raised there. There's no head starting to them. I should also say this work is cool. So if you have any jobs for Jess, I'm sure she would entertain it. And this work was really conducted with her technician, Marlene, who's also looking for grad school opportunities. So Jess and Marlene really put quite a bit of work into this in a site that was pretty unfavorable. Even though my kids loved it, my zoo animals loved these spotted turtle work more than any other work available. I don't know why. I think it was the age, but you can see them down there. We took them out a couple times. So Jess was able to quantify these home ranges and thermal ecology across a few years. This is just the minimum convex polygon data. So what Jess saw was that, like these other turtles, there were seasonal differences in their home ranges, but there was no difference in heavy metals. So the heavy metal accumulation varied, and we'll talk about that in a bit with the turtles, but it had no measurable effect on their home range sizes. And what Jess saw was, again, a little bit different. The ornate box turtles kind of hefted in the spring, blanding in the summer, and then you take this other semi-aquatic, tiny little turtle. Spotted turtles are tiny, the size of your fist. And they emerged from the spring and just started heffing it, even in March. So they're kind of this typical cold weather turtle, I would say, where they can tolerate a little bit colder temperatures. So we saw their home ranges really increase in the spring. They slowed down in the summer, and then they started again congregating into these locations in the fall. And what we thought would happen was they would have these kind of communal brumation sites, and they kind of did, where they collectively brumated. And we thought they would follow the same pattern in the Blandings turtles, where thermally they would move up and down that soil column and effectively just try to stay not freezing. But they seem to be a little bit more tolerant to this. So Jess did it even deeper. She buried these things about 4 feet down. And what you're seeing here is the dark color that's really wavy. Those are the turtles. And the yellow that it's basically right on, that's the air temperature, just ambient air. All the other ones, the water and all the depths, fall in that dark line that goes in the middle. So for some reason, in the wetlands that we're looking at, the temperature in the water and then 4 feet down was very similar. It's probably mostly just saturated soil. And the spotted turtles could care less because they effectively stayed at the surface. Jess would see them under the ice, just sitting there. And when the ice melted, I think they were basking, and they were just hovering around this zero temperature. They never got below freezing, but along the whole time period, they were still pretty active, it seemed like, just not active in how we thought. We thought they would be active in the soil, moving up and down and go through some sort of estivation, but it didn't seem like they did that at all. And they're the smallest turtle. Blandings turtles can get big. And the hatchling Blandings turtles, the head started ones, were about as big as the adult spotteds. And they still did this sequence that was very different. So even though you can look at them and say, oh, it's just an aquatic turtle, whatever, they do these very different sequences, and when they're active and when their thermal ecology changes throughout time. So it's pretty cool stuff. So to summarize this part, with respect to management, we can now tell the DNR and all these agencies that are managing these populations, in the Midwest especially, that their seasonal ecology changes. It's pretty similar to other populations of these species. It's just smaller home ranges in general. But this data was really useful to the managers. They told me personally that they implemented it. They funded the Natural History Survey to expand on it, which is pretty cool. And when we look at this behavior of them, you know, they mimic these non-at-risk species. So if you look at painted turtles or other ones that are really more robust, we see that they have a similar kind of behavioral ecology. The head-started turtles, the ones that were chock full of heavy metals, didn't matter. They kind of just plowed through with the same sequence as the other ones, which I guess is encouraging to see. I hate that we have to do it, but at least we know that behaviorally they're functioning pretty well. So with respect to this conservation physiology now, we examined all these blood chemistries. We looked at the hematology. In this case, I'll present mostly the HL ratios. The heterophil, the leukocyte ratios is a really good way to examine the long-term stress of vertebrates. And it equates pretty strictly to cortisol levels. So if there's low HL ratios, that generally means that the cortisol and the stress levels of these turtles is a lot less. There's not a ton of data on the physiology or the stress of turtles to begin with, you can imagine, especially not sea turtles. So it was hard for us to compare, but when we could compare, these things did pretty well. We put into our linear mix models. There's our blood setup. It's a couple crazy days of trying to dig up turtles from the deep mud as you try to collect blood and a bunch of other metrics here. And you can see the different kind of profiles of white blood cells. Reptiles are a little bit different. They have heterophils instead of neutrophils, and the white blood cells generally look different, right, Jess? Kind of ish. But when you get good at it, you kind of can get these different ratios, and they all tell you these different mechanisms of stress, which is pretty cool. So what I'll show you here is a series of blood chemistry metrics. So from sodium and chloride and calcium, these are these inorganic osmolytes that effectively influence like protein and kind of outside the cell functions, salinity gradients, that kind of stuff. When you look at the organic osmolyte, the only one we measured was blood urea nitrogen, and that's this kind of intercellular water balance thing that's pretty important. And then there's a host of them that deal with either metabolism, a body index and glucose, or kind of diet and body condition focused. So all these different blood chemistry variables can tell you different things about the physiology of these turtles. So what we did effectively was just ask really basic questions of what's influencing these across time or space. So in the case of the ornate box turtles, we looked at whether or not body condition had an influence on these variables. Some turtles can have less fat, or they're bigger or smaller. We looked at if there's differences in sight. And then what we really saw was influencing things were the temperature, basically seasonality. So the body condition of sight, there's no significant difference in terms of these blood chemistry variables and what was happening with at these different sites or different turtles. But once the temperature changed, we saw these variables kind of fall in line with that. And the little data that we can see with other studies show that this was really pretty consistent with other species. They change seasonally, especially in areas like Illinois, where they have to go through some sort of estivation. Not the spotted turtles, but other ones will go through some sort of estivation. And you can see these variables would track through time, where they would change across time as you'd expect based on what they had to do to maintain their body in freezing temperatures. So it's pretty cool to see that we were picking up these signals of changing through time. With the Blandings turtles, we had this unique opportunity to not just look at the turtles that we had started and released, but there are some there for several years that survived for 4 or 5 years in this environment, and we're able to take blood from those. So we weave that into the analysis. We asked whether or not there is an influence on these blood chemistry variables based on not just season, that's kind of the temperature variable, but whether or not they're formerly or recently released. And what we saw for the most part, everything but chloride was not significant. So the ones that were head started, released right away, we tracked them a few times, took blood from them several times throughout the summer, several times for these recently released ones that were 4 or 5 years old. There's no measurable difference. So right away, they're picking up on the environmental cues they needed, and they were adjusting their physiology, even though they were head started and released a month earlier. They're just ready to go. And we did see influences of season, and the non-significant ones were pretty robust, but the ones that were significantly changing across seasons, the sodium and the chloride and the glucose, made sense of why they would. It was all in this kind of preparation for hibernation. So even though their head started, they're doing what they needed to across time, which is good to see. Then when we look at these HL ratios, again, the lower ratios would equate to kind of these less stressed individuals. What we saw when we looked at this, we can compare it to these recently released ones. Those are our turtles that we had started, and the FR, the formerly released, those white ones. So we didn't see a big difference in the HL ratios with respect to the head started turtles versus the ones that were released formally a few years earlier, which is good to see. The stress levels were similar, no significant difference, no predictive ability there. What we did see is that these change seasonally. The stress levels of the turtles change seasonally as you would expect. There's not a ton of data on this, but it seems like it's consistent with other species that we can find. And the HL ratio values are consistent. They're pretty low. There's no indication that these things are stressed, even the ones that were head started and thrown out into this wetland. So the long-term stress of these populations seem to be good. So whatever they're doing in the head starting world seems to be working in terms of providing species that survive well and adjust well physiologically and hematologically, I guess, as well. So what Armand did as well is look at these hosts of other white blood cell types, and the same pattern follows, even with the HL ratios. If you break up the eosinophils and basophils, all these different white blood cell types, they follow the same pattern where, in general, these formerly released ones aren't less stressed than the ones that were just dumped off in the wetland, and across time they change. So we're seeing this kind of congruence across these populations, which is good to see. So what Jess did then was focus on these heavy metals in spotted turtles, and she examined a suite of them. And what Jess was able to do is look at this loosely, this kind of bioaccumulation of metals. So you can see the metals on the left and the sediments and plants and invertebrates, and you can see there's a ton in the sediments. There's way more metals than there should be, to the point where we wore gloves when we were touching the soil because we were so worried about it. And that accumulated into the turtle blood. So the turtle blood values of these different metals are to the right there. Thallium is one that was unique because it's not found in a lot of turtle blood data effectively. It's not run a lot, but there's a crap ton of it in the factories in Gary. They just have a bunch of oopsies where they release it and pay a fine. And we saw that thallium and the cadmium were worrisome, even though the values aren't super high. They're higher than they should be. And they can do some nasty stuff too. So Jess looked at the metals in particular, and we took them in the spring and the fall, and she was able to show that the metals in the turtle blood were variable across the season. So it wasn't this consistent. If turtle had high chromium in the spring, it didn't mean it had high chromium in the fall. So there's some differences in the metal concentrations across time. But I guess what was good to see was that when you compare these metal concentrations to like the 4 other turtle studies that looked at metals in blood, they seem to fall within that range. And some of those are in pretty nasty sites, so the range is a lot higher. But nonetheless, we weren't on the upper part of the range. So they're accumulating metals. And on average, turtle metal accumulation in the blood is relatively low compared to other turtles. But sometimes there could be a turtle that is just jacked full of it, and we have no idea why. But we had to take into account to our analysis because there's a lot of variation in the metal, and you can see it there in the standard deviations. So what Jess was able to do then was take these same blood chemistry variables, and she asked whether or not they differed across site or month or the concentration of metals. What was interesting is that much like these other turtle species, they follow the seasonal pattern. So if you look at it by month, their blood chemistry changes, pretty consistent in what you'd expect. There's no significant differences for the most part on site, and there's no difference with heavy metal concentration. So it had no measurable influence on the physiology, which is good to see, even though it's depressing that they're accumulating a ton of it. But if you look at all these different osmolytes and the metabolic ones and the body condition, there's no measurable difference. So despite the fact that they're in these heavily industrialized areas, they seem to physiologically be similar and doing okay to other species. What Jess also did was look at these HL ratios, and it's the same tune. We saw some slight differences in the site and month, but the heavy metals had no measurable influence on the stress either. So physiologically and hematologically, they were doing well. It's interesting that we saw the same differences in terms of these monthly seasonal changes of stress that the other species showed as well, which is cool. So behaviorally, they act a little different. Physiologically, the turtles here seem to all be just getting ready for brumation. They're fine. Oh crap, I gotta get ready. They change their physiology, their stress changes a bit, and then they adjust. So it's good to see that these things are kind of moving along. So in summary, what we saw was the blood chemistry is really influenced more by temperature or these seasonal changes, even with the head started or the turtles that were chock full of heavy metals, which is cool. And the stress was really consistent to these kind of well acclimated species already. Populations of that didn't have these same threats showed similar stress levels. And in general, nothing would indicate that these turtles are really stressed long term, despite all the challenges they have living in these environments. So to sum up everything, it seems like the turtles are okay, despite you would think they're not okay. But if you look at their home ranges, consistent, they follow similar seasonal trends. There are differences across species, which is cool. They didn't freeze. They're able to kind of account for this behavioral change so they can adjust their thermal ecology. Physiologically, they seemed good. Their trends are following something that you would figure would happen. There's nothing glaring. The stress was really good. So even though that we did our best to screw these things up, we still didn't. And I think that's what a couple 100 million years of evolution does. You can adjust to these different things, which is nice to see. But nonetheless, we were able to provide a bunch of data to these different managing agencies. And then the vet world loves the phys stuff. You know, they have none of it on wild turtles. So they were really happy to get these data. Anyway, I'd love to thank you guys and take any questions. Yes, hi.
Did you find any external manifestations of any of the like eyeless patch-linked tumors, anything like that?
No, what we saw generally were very clear predation events, but there's no external, worrisome things. Even when you look at the blood, so part of what you can do with the blood slides is looking at parasite loads. Those are even lower than you think. So it was all good news and not what we expected. Yeah, John.
Joe, how do you feel what's going on with recruitment across the three species?
Good question. It's hard to say. The box turtle populations seem to be stable but limited in where they can go effectively. But we definitely saw recruitment of box turtles. The Blandings turtles, it's kind of marred by this head starting. They released thousands of turtles so far, but they have started to get recruitment. So they started the program, I think, about 20 years ago now, and it's a big part of what DuPage County does. And they found turtles they released from head starting actually laying eggs. So there is new recruitment of Blandings turtles going on. The spotted turtles seem to be rolling. You look at it and you cannot imagine they would be okay. The sites are awful. Calumet Prairie is a disaster. And there's so many spotted turtles, young, old, whatever you want. So they're doing the best, I would say, out of the bunch. Blandings turtles are supplemented heavily, but they seem to be picking up pace. And as far as we could tell with the box turtles, it's a more older population, but we found Jenny Wren found hatchlings. She dug up a hatchling for John and brought this little baby ornate box turtle. So there is recruitment going on, but I don't think anyone's really dove into, the analytics of it. Hi.
Hi, great talk. I'm curious again about this heavy metals. So I guess it's a multiple part question. And I apologize for that. So are you able to age the turtles to determine if there's I guess my question is, even in older turtles that have bioaccumulated a ton of these heavy metals, are they still fine? And my other question would be, are these levels of heavy metals that are in the ecosystem, would those be toxic to mammals like us? And if it is, and they're unaffected by it, does it have something to do with their slow growth and their physiology?
It's a good question. I don't know if I know the answer to any of those, sadly. The first is the aquatic turtle aging is difficult. The turtles have growth rings on their scutes that sort of could work, but when you look with aquatic turtle, they get worn down a lot. So all we could really say, they were all adult spotted turtles. What we could say is that the turtles that had higher heavy metal loads for some metals still looked physiologically and stress similar. The second question, I don't know if anyone's even looked at that. We haven't dug into the heavy metal concentrations to see if they're toxic enough. But what jarred me, when I talked to someone about the loads in the sediment, and they said, don't touch the soil. So I don't know, there's not a whole lot of blood. We really this is new data, like weeks old. So we focused on the turtle stuff first. What could we find in relation to turtle blood? And that was minimal. So we haven't really extended past the toxicity in mammals or other species, but we'll do it. It's a good question. Sorry, I don't know the answers, but.
So great talk. I apologize for having another multi-part question.
No, I.
It's all about the ornate box turtle though, so it's kind of localized.
Yeah.
So are those the same box turtles people keep as pets? People keep as pets?
People do take them as pet. You're probably talking about the eastern box turtle. That's a more widespread, mostly kind of forest adapted. There's a three-toed box turtle that is more forest adapted. So ornates are definitely in the pet trade because they're prettier. You know, the pattern is more ornate. So yeah, I think the answer is yes. They trade the hell out of them. Probably less so because there's less of them, but they're definitely still have a threat for you know, status of that.
So that is part of the harvesting you're talking about.
Yeah. Yeah, it's harvesting for pet trade in the US. That's probably one of the number one things. And I think what's overlooked a lot is Southerners eating turtles. You know, there's very few restrictions on turtle, cat, and herps in general. They call salamanders down south spring lizards, and if you got a fishing permit, you can get as many as you want. Endangered, what they don't care. And I think turtles have that similar kind of restriction, even though there might be some laws in place. I don't know how implemented they are. And it's hard to catch folks that can put a line in and get a you know, soft shell turtle in a second. That's it. So I think we can't quantify the harvesting of turtles for meat in the US very well. Outside of the US, it's a huge problem for real. And the pet trade is one that is also hard to kind of quantify. And I don't know if there's a ton of resources or interest in quantifying it.
Okay, so this is like part 3 of the question. You mentioned the fecundity rate. What is it like for turtles? Like for that turtle in particular?
Okay, so for ornate box turtles, most of the turtles, I would say, they take 10 to 15 years before they become fecund.
Oh my God.
And then it could be, as far as we know, yearly, maybe biannually. Yeah, right. I mean, it's sure. And then after, you know, the box turtle can live to 90 years, but I don't know when they stop. They just say, screw this. You know, I'm too old. So they lose that fecundity at a certain point. So there's a window that you say is small, but it could be 20 years where they're reproductive.
There's no captive breeding for the ornate box turtle, even though it's pretty popular.
Not in Illinois. The populations seem to be more robust than blandings and spotted turtles.
Okay.
So, and again, when you're looking at herp stuff, me and Sarah care, and like, that's about it. You know, like they're not hugely popular. They're not charismatic. They don't bring in a lot of money. They're hard to find. So I don't know if we really know the status of ornate box turtles in Illinois as much as we should. I know they're in decline. They're definitely at risk. I don't think they're even endangered in Illinois. They technically probably are threatened, right? So it's an issue that we just don't know.
Okay, thank you. I might talk to you more later.
Yeah.
I saw some Phragmites in the photographs, and I was just curious how you'd comment on the distribution.
Yeah. Jess can talk a lot more. The spotted turtles, that's where the Phragmites sites, they loved it. Not at first. They congregate in the ephemeral wetland at that site, and the other site didn't have a lot of Phragmites. But Jess spent hours, days, you can argue, walking through Phragmites, seeing turtles, maybe traversing through it. And the Blandings turtle hatchlings liked it, I think, as well for cover. But it's hard to say that because that's what was mostly at these sites. So did they like it, or is that what they had to offer? But they dealt with it at the very least.
Joe, I wanted to ask you about, so how possible is it to mark these turtles for the long term,
Oh yeah.
for
that's-
multiple years? Can you do that?
Oh yeah, very easily. You can do basically shell notching that last for decades. And folks have found box turtles 90, 100 years after someone marked them. So those are pretty reliable. Aquatic turtles get a little more dings in their shell. But it's still, yeah, definitely. You could tag them pretty easy too. So if you can find long-term money, if you have any, I'll take it. But we can totally mark these things for decades. And that's the only reason really we can study them long-term, to look at their age and their fecundity, is that people have done it. They've rustled that up.
Hey, thanks for your talk, Joe. So I think it's really cool what you guys have done in terms of collecting these physiological metrics from blood. And so I was just curious from a field sampling perspective, when you're looking at stress and glucocorticoid levels as a proxy, when you're sampling, do you have to like process the animal or take those blood samples within a certain amount of time after capture versus processing them, like capturing a lot of turtles and processing them all later, just based on whether or not those glucose levels can like build up in the blood for a certain time after capture?
That's a good question. For the blood physiology, there's a window, but it's 4 hours or so. So it's not terrible. The beauty of the blood slides is it's a long-term stress measure. So you don't need to rush and take blood real quick. And if you're taking blood from a turtle, they are great at not giving up their blood. So Leanne is a master at it. She can hit the vein usually in a couple tries. And the box turtles especially, they can close up. But the beauty of the HL ratios and the slides is that it could be days because it's a long-term measure. That the leukocyte profiles take a long time to change. So that's why we kind of switch to that rather than the cortisol. The blood chemistry though, you don't want to leave it in the trap overnight. You have to account for that. And the phys people will get a little testy about that. But their sample sizes are also like 5. So when you present something with 20, they're like, yes. And they tend to care less that it took us a couple hours. But we haven't tested it directly to see if we take blood right away and wait, and then take it if the blood chemistry changes. And glucose is one that we oftentimes don't make a lot of inferences on, because if the turtle just ate or is going through something, that changes real fast. The other inorganic and organic osmolytes don't change as fast, so we weren't super worried about it.
Looks like there's no more questions, so let's have a round of applause for Joe.
Thank you, guys.